we are moving into a more realized idea of photosynthesis. So in the thylakoid membrane is where we are starting. Remember that thylakoid membrane is that kind of internal stack of membranes inside of chloroplasts. Chlorophyll molecules are organized with other molecules and proteins into complexes called photosystems. And these photosystems are where our light reactions take place. So, specifically, what is a photosystem? I'm just gonna make this really big. I got tired of pushing it with every slide last time. Okay. All the way back up. So, photosystems. I'm just gonna are comprised of a reaction center with several light harvesting complexes. So all those chlorophyll molecules and all those other associated molecules and proteins are organized into a reaction center complex and a light harvesting complex. So we're going to talk about how both of those work, starting with the simpler of the two, the reaction center complex. Reaction center complex. Now, a reaction center complex is an organized association of proteins. So organized proteins goodness gracious. Holding a special pair of chlorophyll A molecules. Nope, that's not going to work. And a primary electron acceptor. So we've got this reaction center complex, and it's just a bunch of proteins, and those proteins hold a special pair of chlorophyll A molecules and a primary electron acceptor. And that is, as you may guess, where a bulk of the first reaction in the light reactions take place. And then we have our light harvesting complexes. as we talked about yesterday with our pigment molecules, they are the best at getting that light absorbed. So our light harvesting complex is just various pigment molecules bound to proteins. And the way it works is if you were to look at a photosystem, you would have all of these light harvesting complexes, so all of these pigments and proteins surrounding in the center a reaction center complex, sorry, with its two special chlorophylls and that primary electron acceptor. The light harvesting complex acts as an antenna for the reaction center complex. So it acts as an antenna for the reaction center process complex. I don't know why I keep saying process. So the light harvesting complexes kind of suck up the light energy and channel that light energy into the reaction center process complex. Goodness gracious. Because when a pigment molecule absorbs a photon, this is in the light harvesting complexes. So if one of those pigments in the light harvesting complex absorbs a photon, the energy is transferred from pigment to pigment within the light harvesting complex. Let's say energy, yeah. Transferred from pigment to pigment within the light harvesting complex. So it would just be like if this is one of our pigment molecules and this is another pigment molecule, and this is another pigment molecule, if this one gets excited, one of the electrons will go up in energy level, but that's pretty unstable. So it'll immediately drop back down. And when it drops back down, it will transfer the energy to another one. And the same thing will happen with another one. And that electron or that energy is going to be passed from pigment to pigment to pigment within the light harvesting complex. Now, we're going to talk about that special pair of chlorophyll A's in the reaction center complex. So, special pair of chlorophyll A's in the reaction center complex that's where like the big idea of this kind of process happens. Their location enables them to use energy from light to transfer the energy of the electron to this electron acceptor. So this special pair of chlorophyll A molecules in the reaction center complex are positioned or is positioned to, when excited, transfer high energy electrons to a primary electron acceptor. Transfer electrons to the primary electron acceptor. Now, a primary electron acceptor is a molecule capable of accepting electrons and becoming reduced. So, molecule capable of becoming reduced. So, we've got our special pair of chlorophyll A molecules, and they are positioned just right into that reaction center complex. When they get excited, either from being hit by light directly or from having energy transferred to them, we'll send those electrons up to the primary electron acceptor, and that is what gets the ball rolling. So, yes, kind of our AP big idea here, I know it's kind of wordy, is that the solar-powered transfer of electrons from the reaction center, chlorophyll A, to the primary electron acceptor is the first step of the light reactions. So first step of light reactions is the solar-powered transfer of electrons from the reaction center chlorophyll A pair to this primary electron acceptor. So when that primary electron acceptor finally gets the electron it needs, we will kind of move into our next step, which we'll really talk about tomorrow. So I'm going to talk about a little bit of the difference between photosystems, because there's two types of photosystems. And kind of one more jab at our good buddy, the redox reaction. So when chlorophyll is excited, and that electron is captured by acceptor, that is a redox reaction. Because our primary electron acceptor is being reduced, therefore our chlorophyll A molecules are being oxidized. Because you can't have a redox reaction with just a reduction, you also need that oxidation. So there are two types of photosystems. Or two photosystems. They are named photosystem 1 and photosystem 2. But when we write them, we always write them photosystem 2 and photosystem 1, or PS2 and PS1. If you can keep your brain from calling it a PlayStation. So here's the thing. They are named after the order in which they are discovered, not the order in which they take place in photosynthesis. So, photosystem 2 happens before photosystem 1. So they're named after discovery. But photosystem 2 happens first. That is why we always call them photosystem 2 and photosystem 1. Or PS2 and PS1. So, the difference between the two, there's three differences between the two. They have characteristic reaction center complexes. So they're reaction center complexes. Reaction center complexes are slightly different. They have a particular kind of electron acceptors, different electron acceptors. And they have slightly different chlorophyll A pairs. Sorry. Slightly different chlorophyll A pairs. Yeah. So, talking about their reaction center complexes, the reaction center in PS2, reaction center complex in PS2, you can actually write that all out, but is also called P680 because it absorbs mostly wavelength 680. Wavelength, I'll write out wavelength. And that is really, really red. That's the wavelength that it functions highest at. And the PS1 is P700. So very slight difference, and that is a far red. So one more thing before I actually draw it out. They are nearly identical, especially in their associations with different proteins in the thylakoid membrane. So they look really, really, really similar. They're just slightly chemical differences. the proteins, okay, yeah, and it is the slight differences in the proteins that they are associated with that affect electron distribution. So they look they look similarly, and they are associated with proteins similarly, but the proteins that they are associated with are slightly different because the way that electrons are distributed from them are different. And that is kind of the goal of tomorrow, is to talk about electrons. Yes. So, let's just draw one. Drawing always helps. So, if we've got our thylakoid membrane, goodness gracious, thylakoid membrane. This is the stroma up here, and this is the thylakoid space. Having this kind of spatial awareness will help you whenever we diagram photosynthesis later in the week. So, we have our thylakoid membrane, our stroma, our thylakoid space. So, because we are bisecting this, essentially it would be like a little sphere. Imagine it like a corn dog. That'll probably help. So with the light harvesting complexes being the like breaded part, and the reaction center process being the actual like hot dog part. So that is what I'm attempting to draw a bisection of. Kind of looks like a pumpkin. Okay, that's what we're looking at. I'm going to color code this to help it a little bit easier. So the this box right here, this is our reaction center. And this out here is our light harvesting complex. So we have our special chlorophyll A's, if you can see that. Special. Our special chlorophyll A pair that exists in our reaction center, along with our primary electron acceptor. Let's see. And then in our light harvesting complex, we have our pigment molecules. You can't see that. Barely, at least. That's our pigment molecules. This entire thing is our photosystem. If that didn't make it super, super messy. So, what happens me get this orangey brown color. If this is a photon, and these brown arrows are the transfer of energy. A photon will hit one of the pigment molecules in the light harvesting complex, and it will bounce around until eventually that light hits the special chlorophyll A pair in the light harvesting complex, and that special chlorophyll A pair will send those electrons to the primary electron acceptor. And this is what happens in photosystem 2 and photosystem 1. So this process is exactly the same in the both of them. It is what the primary electron acceptor does after the fact that differs between photosystem 2 and photosystem 1. So in a thylakoid membrane, there would be a photosystem 2, a little bitty electron transport chain, and then a photosystem 1. And we will diagram that out again tomorrow. So far, does anybody have any questions? I know we had to build up a lot of physics and a lot of light stuff before we got to this point. Everybody doing okay? Cool. There is an exit ticket on Canvas. It didn't take as long as I expected us to. But there's an exit ticket on Canvas, and if you have any questions about that or about this, please just let me know.